In this session at Medical Sciences, we are going to take you through what we call bacterial growth curve. And in this session, we are going to see the requirements for the bacteria to grow and different phases in which the bacteria passes through after being cultured in the media. And as we begin, please remember to subscribe if it is your first time. If you haven't subscribed, then like the video so that we can continue to make more others like this. So with bacterial growth curve, you want to see phases on which the bacteria passes through before it grows and then it dies. So, and we know that bacteria grows by the process known as binary fusion, binary fission, whereby if you have a single bacteria with a DNA, which is always chromosomal DNA, this one-celled bacteria, after being cultured in either in a media which can be solid, semi-solid, or liquid, after adapting to the environment, it starts to divide by binary fission, whereby one bacteria increases in size, and then this bacteria is going to increase in size. It increases in size, and the DNA divides into two. This circular DNA, the chromosomal DNA, divides into two, and this single-celled bacteria develops a furrow. It develops a furrow. That is the line of weakness where it is going to divide. Then at the end, it is going to give into two daughter cells, whereby it is going to give two daughter cells, with each having its chromosomal DNA. So this process, this is the process we are calling binary fission, whereby the bacteria are able to reproduce or to increase in number by one cell giving rise into two daughter cells. And this is what concept you are calling binary fission. So these two daughter cells which have been given also go ahead to give more two. Also this one goes ahead to give two. So at the first we had one, we got two, then we get four, and off. So it keeps multiplying in doubles. So here we are able to get more two after this replicating. Here we get also more two bacteria with the DNA, each having its own structural DNA. And this process is what we are calling binary fission, and it occurs by doubling of cells. So the bacterial cells keep doubling. That is what we call binary fission. And the time taken by a single cell of a bacteria to produce two daughter cells is what we call generation time. This is generation time. It is the time taken for a single bacteria to give rise to two daughter bacteria cells. That is what we call the generation time or doubling time. You can call it generation time or doubling time. The time lag it takes for a bacteria to increase in number or to give rise to two daughter cells is what we are calling generation time. So when we have a media, for example, a culture plate, you have a culture plate and you come, you streak the culture plate with the organism of interest for which you want to isolate, maybe by streak method, and you incubate it at required temperature, pH, then light. You can include with required nutrients. then with required osmotic pressure. All these are the requirements for the bacteria to grow. So for this cultured bacteria, what does it need? It needs nutrients, like the agar base containing like nutrient agar and peptone. Then you need temperature. You must incubate at the right temperature, out of which we know most of the pathogenic bacteria grow at 37 degrees. Then you need the appropriate pH. You need the appropriate light for those which are phototropic. Then you also need the osmotic pressure for those which are halophilic. So these are some of the requirements for the bacteria to grow. And apart from those ones, also they have to adapt by coming up with necessary enzymes. So as we begin different phases, these are some of the requirements we need for the bacteria to grow. That is nutrients, we need light, we need temperature, we need pH, or optimum pH, we need light, and we need osmotic pressure for the bacteria to grow. So after culturing it on this agar plate, we are talking about this agar plate, there is the time, there is the first phase, we call it lag phase. And this lag phase, it is the time taken after inoculating the bacteria on the agar. The time it takes to increase in number is what we are calling the lag. So here we don't see any increase in number. That is the lag phase. Then it is followed by another phase known as the log phase, or you can call it exponential phase, where there is increase in bacterial number. Then after this exponential phase, we come up with the stationary phase, whereby there is no increase. Then we end up with the decline phase. So these are the phases we are going to look at under this bacterial growth curve, where the first one is lag phase, followed by log phase, followed by stationary phase, and lastly decline phase. So this one, we are calling it log phase. This one we are calling it stationary phase. Then this is decline, or you can call it death phase. This log phase, you can call it exponential phase. So these are the four phases under bacterial growth curve, whereby the first one is the lag phase, the second one is the log phase, the third is the stationary, and the fourth is the decline phases. So after knowing these different phases, so let us explain each phase one by one. And we are going to begin with the lag phase. So number one is the lag phase, which we said it is the phase, or it is the time between inoculating the bacteria on the culture media and the time it takes to start multiplying. And that is what we are calling the lag phase. And here, in this lag phase, there is only increase in size. The bacteria is increasing in size, but there is no appreciable increase in number. No appreciable increase in number. So there is no increase in number of bacteria. So the bacteria is remaining the same, but what is happening, it is increasing in size. And as it is increasing in size, it is adapting. Here, there is what we call adaptation. It is adapting or acclimatizing to the new environment. How? By adjusting temperature on which you are incubating, by adjusting to change in pH, by adjusting to other atmospheric conditions like oxygen. But at the same time, this bacteria is also making necessary enzymes which are required for metabolic processes. So during lag phase, there is protein synthesis in the bacteria. That's why we see there is increase in number. And this protein synthesis is bringing about formation of the required enzymes which are required to carry out metabolic processes during bacterial growth. So that is what we can see under acclimatization. It is getting adapted to the new environment, getting adapted to the temperature, and processing required materials like enzymes to carry out metabolic processes so that it can start increasing in number. And at this point of lag phase, the bacteria is metabolically active. Here the bacteria is not dead, it is metabolically active. That's why we see there is formation of new enzymes that are required. Then from there, after getting the necessary enzymes to carry out metabolic processes, after adapting to the environment, and after increasing in size, then we go to the second phase, which we are going to call log phase. And this log phase, you can also call it exponential phase. It is also called exponential. And at this stage, the bacteria has already adapted and there is necessary nutrients, there is availability of nutrients, availability of nutrients are present, and the bacteria starts multiplying, whereby this multiplication, as we have said, is by binary fission, as we have explained it from the beginning, whereby if you have one bacteria, it gives rise to two, two gives rise to four, four gives rise to eight, eight gives rise to 16, and 16 gives rise to 34. So there is doubling of bacterial cells during log phase. That's why we call it exponential. There is increase, and the generation time differs from bacteria to another. As we said, the generation time is the time taken for one bacteria to give rise to two, two to give to four, four to give to eight. That is what we call generation or doubling time, which differs from one bacteria to another. So, meaning during this log phase, the rate at which the bacterial cells are being born is far greater than those which are dying. So the birth is greater than the death, those which are dying. That is under log phase. And it is at this phase where bacteria are very sensitive to, so in this stage, the bacteria are very sensitive to hostile environment, for example, like presence of antibiotics and other antimicrobial agents. And it is at this stage where there is good staining, where there is, you can easily stain to differentiate different bacteria by their morphology. That is during log phase. Then after them multiplying, of course, they are going to reach maximum. They multiply until they reach maximum number or maximum density, whereby they are going to deplete the nutrients, they are going to cause change in pH, they are going to start producing exotoxins, and some cells begin dying. And this one ushers us into the third stage, which we are going to call stationary phase. And this stationary phase, it is at this stage whereby the number of bacteria being born, the birth rate, is equal to the death rate. So there is almost no more increase, but there is little, because we see some are being born, others are dying, but the number equal, and that's why we are producing a stationary phase. And this stationary phase is brought about due to increase in cell density of the bacteria. So due to increase in the population of the bacteria, they start competing and deplete. There is depletion of nutrients. There is depletion of nutrients due to increase in the number, and because there is increase in cells, and the increase in cells and there is production of exotoxins, they lead to change in pH. Change in pH. They can lead to depletion of required environment like oxygen and water, and this changes due to depletion of nutrients, change in pH, oxygen and water, and the change in pH is brought about by accumulation of toxins. Accumulation of toxic metabolic products. So when the toxic metabolic products accumulate, lead to change in pH, then oxygen is depleted if it is required, water if it is required is depleted, then there is also depletion of nutrients. So the bacteria, they stop multiplying, and some are being killed by these toxic products which are being formed. But the good news, we are saying the rate of birth is the same as death, and that is stationary phase. And it is at this stage whereby we see the bacteria start producing endospores, so that they can survive in this harsh environment. They begin producing exotoxins and endotoxins, which are destroying other cells. So these are some of the things which are causing the birth number to be the same as the death number. That is depletion of nutrients, accumulation of toxic products which lead to change in pH, and lack of necessary physical requirements like oxygen, like osmotic pressure, leading to some of them dying, but the number of deaths being equal to the birth, and hence remaining stationary. And it is at this stage whereby the bacteria is adapting by producing endospores and producing exotoxins. And here the staining becomes variable. They lose their staining characteristic. So finally is the last phase. And this last phase, we are calling it decline phase. And during decline phase, which is phase number 4, and decline phase, we are also calling it death phase. So here the bacterial cells starts to decrease in number. There is decrease in the number of bacterial cells, and why are they increasing? It is because the death rate is far greater than the birth, those which are being produced by binary fission. And that's why we see there is a decrease in the number of the bacteria. And you can ask yourself, what is causing the number of bacteria to decrease? What is causing much death? It is due, of course, to depletion, complete depletion of nutrients, and continuous accumulation of toxic metabolic products. And these toxic metabolic products kill the bacterial cells, kill these cells, hence leading to the number of deaths greater than birth rate. And it is at this stage whereby the viable count here, the viable count is less than the total count, the total bacterial count, whereby the viable count is the number of bacteria that are living or that are metabolically active and are able to multiply, whereas total count includes both which are living and those which are non-living. So at this stage, the viable count decreases, but the total count remains constant. So this is the total count. Then this is the variable count. These are the ones which are living. Then this one combines those which are living and those which are dead. That is what we are calling the dead. The variable count being less than the total count. So this is what we can discuss with you in this session, whereby we have looked at the requirements. We have looked at the different phases, that is the lag phase, log phase, stationary phase, and decline phase. And we have gone ahead to explain each phase, how it is brought about. Hope you have enjoyed this video. Please remember to subscribe, like the video, and share to many others. Thank you so much, and may God bless you.